Now, normally this lecture isn't included in the cell division unit, so I'm going to get into DNA replication, and usually this is taught in a molecular genetics unit, or basically a unit about DNA. The problem, though, is after we teach you about the structure of DNA, and then we teach you how DNA gets replicated, and we teach you how we can get proteins, or protein synthesis happens regarding DNA, you get those two processes, replication and protein synthesis, all confused. So I decided to pull replication out of the next unit, the molecular genetics unit, and put it in this one. Since we're talking about cell division, and we've already mentioned that during S phase, right here, is when that DNA is getting replicated. And another reason why is that molecular genetic unit is a monster. There's a lot that we go through. So I figured if I pulled replication from that unit and a DNA technology called PCR, then that would make that unit a little more manageable. So we're actually going to look and see what happens to DNA whenever it's being replicated. And again, it only gets replicated at one time in the cell cycle, and that's during the S phase. And that's why I've indicated that right here. DNA can change forms throughout the cell cycle. So we see sometimes DNA indicated like a double helix, and sometimes we see DNA in chromosome form. Now, these different forms of DNA sometimes are referred to as uncondensed forms of DNA. We can also say that DNA is in chromatin form. And in order for DNA to be replicated during S phase, and also in order for it to code for proteins, like for transcription to take place, it has to be in chromatin form. When DNA is coiled up or condensed, then we can't replicate it and we can't transcribe it to get a protein from it. The only time DNA ever coils up, as we can see happening from here to here, or condenses, is whenever it's time to divide up the DNA. So mitosis is occurring. It's easier to move and divide up these molecules of DNA whenever they're condensed into chromosome form. Throughout all this time from here to here, DNA is uncondensed. And then we learn that it condenses up right here. So it condenses in prophase and it stays condensed until it's divided up in mitosis. Before we can go through DNA replication, we need a quick review of the structure of DNA. I'm not going to take the time to read through this information, but we've already talked about in the biochemistry unit. We hit it hard in pre-AP. So if you need, just read through this information to re-jog your memory about the structure of DNA. Probably the most important information that I want to highlight are the ends of DNA. Remember that when we have an end and it's close to that phosphate group, then we call this the 5' end. And to remember that, I like to think of, we can take this end with the phosphate group, so there's a P there, and it's easy to turn that into a 5 because a 5 has one hump, just like the P has one hump. So you might remember that if you see a phosphate at the end, then this is the 5' end, and see that this carbon atom of the sugar is technically this end, so this is called the 3' end. And these strands are anti-parallel, so that means that this is the 5' end, and you can see it's close to that phosphate group there, and then this is the 3' end. And it's important to understand that there's 5' and 3' ends for each of these strands when we're going to go through replication. Before we actually get into DNA replication, I want to talk about the 3 models that outline how DNA could replicate itself. There's the semi-conservative, the conservative, and the dispersive model of replications. Now, the one that's actually accepted as being correct is the semi-conservative. I'm going to go ahead and mark through the names of the others so you know that these models are no longer accepted. At one time, though, we did not know which model of replication was the correct one. And it took the work of 2 researchers, Messelson and Stahl, to actually figure out that the semi-conservative way of replication was the accurate one. In the semi-conservative model, it's basically stating that we have some parental DNA, and I'm going to mark both strands with a little star here. When this strand of DNA is replicated into 2 strands of DNA, which we see here, each new DNA molecule is going to be made of one of the parent strands. So here's one of the original parent strands that I marked here, and a newly synthesized strand, and that's indicated with this blacker coloring. So again, in this second DNA molecule that was formed through replication, we get one of the parent strands, so this one right here, and then we get a newly synthesized strand of nucleotides. With the conservative model, it was believed that this parent DNA molecule was fully conserved. It was kept completely intact. And then the machinery just built a copy of it all out of new nucleotides. So that's why it's called the conservative models, because we conserve the original. If you think about this first model that we talked about, we kind of conserve the original. Half of it ends up here and half of it ends over here. So that's the semi-conservative model. In the dispersive model, it was thought that some sections of the DNA were conserved. So like from here to here was kept intact. And you can see that right here. But the black sections were newly synthesized from free nucleotides. Now, to prove which model was correct, Messelson and Stahl set up this following experiment. AP loves to ask you questions about experiments like this. Now, technically, the Messelson and Stahl experiment is not one that you need to have memorized. In the next unit, there's going to be specific scientists that you need to have memorized. You need to memorize their contributions to the discoveries of DNA. These guys weren't listed as one of them. But still, they can give you this information, and you have to be able to come up with conclusions based on the data from the research. Also, research like this emphasizes why they want you to have memorized some of the elements that make up different molecules. So, for example, for Messelson and Stahl to set up this experiment, they had to know that DNA was made of carbon, hydrogen, oxygen, phosphorus, and nitrogen. Something else that they had to have some knowledge about was isotopes. Now, there's two different isotopes for nitrogen. There's nitrogen 15 and there's nitrogen 14. And 15 is more dense than 14. Now remember, the bases of DNA, adenine, guanine, cytosine, and thymine, they contain those nitrogenous bases. So when DNA is being synthesized, if you only give the cells access to nitrogen 15, then that's the only thing that they can build their DNA with. We just add nitrogen 14 to a medium where there's cells that are actively dividing and replicating their DNA, then the only thing they're going to be able to use to make their DNA is this nitrogen 14. So here's how they started this experiment. So they started growing bacteria in a medium that only had the nitrogen 15 atoms in it. So if these cells are copying DNA, making new molecules of DNA, then all that DNA is going to have is those nitrogen 15 atoms. We can remove that DNA from the cells, we can spin it in a centrifuge, and I'll show you one in class. And when we have molecules in a tube and we spin them, then these molecules will actually separate out into different columns. And the more dense molecules will go towards the bottom. So whenever they centrifuged all this DNA from these bacteria cells, then they got these bands at the bottom of the centrifuge tubes, showing that they had some pretty dense DNA. Then what they did is they took some cells out of this first batch that had been tagged with that nitrogen 15, and they put them in a medium that only contained this nitrogen 14. And they let them grow for just one generation. So these cells copied their DNA once and they split in two, and that was it. They removed the DNA from these cells, and again, they spun it, they centrifuged it. And what happened is the band wasn't so far down in the centrifuge tube. The reason why this is because the DNA that was made had a copy of the original strand and then a newly synthesized strand, and this lighter strand had those nitrogen 14 nitrogen atoms in those bases. And the same with the other one. It had the original strand. The original strand had nitrogen 15 atoms in it. And so whenever we spun them out, they had a density that was less than those DNA molecules that were all nitrogen 15. But they weren't done. They decided to take these cells, these hybrid cells, and let them divide for one more generation. Again, remove the DNA, they centrifuged it, and this is what they got. So some of those hybrid DNA molecules would have separated out to make this hybrid band, just like we saw it here. But then we got a brand new band that we hadn't seen anymore. We got a brand new band that we had not seen yet. And that was because we ended up with some DNA molecules, and they all contained those carbon 14 atoms. And these DNA strands were the least dense, and they separated out to form that band at the top. because all of the nitrogen atoms in these DNA molecules were the nitrogen 14. So the only way to explain these results is if semi-conservative replication was occurring. If the other two types of replication were occurring, then we would not have gotten the bands that we did in the 3 different parts of this experiment. Remember I mentioned that DNA replication and transcription of protein synthesis, they look very similar. So students have a hard time recognizing whether they're looking at replication or if they're looking at transcription. So I wanted to show you a bunch of different pictures of replication. The more diagrams you see, then the better able you will be to figure out whether you're looking at replication problems or transcription problems. So here's some things to look for. So first of all, you'll see a DNA molecule, of course, and sometimes you see many little openings in it, like we see here. Those are called replication bubbles. Down here, I'm showing you a diagram of what DNA actually looks like whenever it's being replicated, and you can see several replication bubbles opening. Now remember, DNA is a really long molecule, and you have several molecules of DNA in your cells. You have 46 molecules of DNA, and that's why you have 46 chromosomes. And we end up with about, it's about 4 billion base pairs is how long all of your DNA is in one of your cells. So it's going to take a really long time to replicate it, and that's why you see several replication bubbles open. So we have replication starting here, we have replication starting here, we have replication starting here. If this just happened where we opened DNA at one end and then we just unzipped it all the way from one end to the other, and that's all that was being replicated at one time, it would take DNA forever to get replicated. So it has to be replicated pretty quickly, and so several bubbles are going to open up where replication is happening along that molecule. Now, here's another diagram of a replication bubble. So they're zooming in there, and you can see things like the leading strand and the lagging strand, and then they have names of some of these enzymes that you probably recognize from pre-AP. We talked about helicase, we talked about DNA polymerase. One big thing that I want to point out, though, is that DNA polymerase, let's think about what the name of this molecule means, because I have to teach you RNA primase, I have to teach you RNA polymerase when we get to protein synthesis, and if you can just think about what the name of this enzyme is, then it's going to tell you what this enzyme is doing. DNA polymerase is putting together, it's making a polymer of DNA. So this is the enzyme that actually puts together DNA nucleotides because we're trying to make some more DNA. We're trying to replicate. So when I teach you about RNA polymerase, then you need to know that's the enzyme that is actually making RNA, and that's during transcription. We do not want to get the two confused. Now, the next diagram that I want to show you is just a diagram where they're zooming in, and you're just looking at this part of it. So it doesn't really look like a replication bubble, but it is a replication bubble. They're just showing you half of it. So I just wanted to show you there's many looks to replication. Sometimes you're shown the entire bubble, sometimes to zoom in to emphasize the names of certain enzymes and so on, then they're just going to show you what's actually called the replication fork. They're just not showing you the back part of the bubble on this diagram. But again, all of these diagrams are showing you replication. Now I'm going to attempt to draw on some of the events that take place whenever DNA is being replicated. And I'm going to start with the very simple diagram that you see here. Now, the players that are involved, the enzymes that are involved, are listed for you up here, and it tells you what each one of these enzymes do. And you need to have a good understanding of the function of each of these enzymes. Before I begin, I also want you to understand that in the nucleus where replication is occurring, there's tons of what's called free-floating nucleotides. There's nucleotides everywhere. So here's a nucleotide, and maybe this one has adenine on it. So there's a phosphate, there's the sugar, and then there's the base, adenine. And this one would have maybe guanine on it. So there's a phosphate sugar, and then the base is guanine. And we have these everywhere. So we have these free-floating nucleotides that we can use to build new strands of DNA. In pre-AP, we learned about this guy right here, and it is opening up the DNA strand. So basically, we're breaking those H bonds. Remember, there's H bonds here, holding the bases together. And helicase, so let's label it. is the enzyme that unwinds the DNA or breaks apart the bases at the hydrogen bonds. Now, further down the line, there's an enzyme, and I'm going to draw it on here. I'm going to kind of fill it in there. And this one is called topoisomerase. Now, remember DNA has a twist to it. It has that double helix structure. Whenever we unzip it, if you go to unzip basically two strands that are twisted around one another, then that's going to end up putting a lot of tension further downstream of where we're unzipping. And I'm going to do a demo of this in class so you understand what's going on here. So topoisomerase will actually cut the DNA and untwist it a little bit. So there's less tension on the DNA strand so it doesn't just break on its own. And then it's going to rejoin those DNA strands. Now also, as we unwind it, we have to keep these two strands of nucleotides apart, because if they come in close proximity to one another, they'll reform those hydrogen bonds. So there's proteins that will bind, and these are called your single strand binding proteins. So they bind to the single strands of nucleotides that have not been separated there, and it's just keeping them apart. Now, the major enzyme that is going to build new strands of nucleotides is DNA polymerase III. Now, there's three, and there's one that I'll mention, and I don't care that you know the difference between 3 and 1. It's just sometimes I think students actually like to know that there's DNA polymerase III, and it's different from DNA polymerase I, and they have two completely different jobs, and it helps them remember the different jobs. Now, when DNA polymerase III goes to build a new strand of nucleotides, it can only build the new strand in the 5' to 3' direction. And that's why I was bringing up that information about DNA, because it's important that you understand what we mean by the 5' and the 3' end. And you're also going to see that DNA polymerase, it cannot bind to a 5' end of a nucleotide. It can only bind to a 3' end of a nucleotide to start DNA replication. So let's add some ends here to our DNA molecule strand. So let's say this is the 5' end down here on this nucleotide. And then for this nucleotide, that's the 3' end. And if we follow this strand of nucleotides all the way down here, we're going to end up, this end has to be the 3' end, because it's going to have to start with the 5 and end with the 3. If this starts with the 3, then this is going to be the 5' end down here. Now, I can't go through what DNA polymerase is going to do specifically yet, because DNA polymerase cannot just hook on to this DNA strand and start replicating it. It has to have a 3' end of a nucleotide to hook on to. But a molecule that can just hook on to a strand of nucleotides and match up bases to it is this molecule here. It's called RNA primase. Sometimes it's just called primase, but I think it's easy to remember that it's RNA primase because it's going to grab free-floating RNA nucleotides and it's going to match them up, creating a section of DNA which is called a primer. Now I'm going to add some bases here. So let's say that this DNA was A, C, T, G. RNA primase is going to add a primer, and it's going to match up with RNA nucleotides. It's going to put C, A, G, and U. Now we have, right here, we're going to have a 3' end of a nucleotide. Because remember, this one would have to be down here, on the end of this nucleotide would be that 5' end, because we always have a 3' to a 5' on a strand of a DNA molecule. So now we have that 3' end, and DNA polymerase can hook on. So let's show DNA polymerase can bind right here, and it's going to move this way, and it's going to start grabbing free DNA nucleotides and attaching them to their complementary bases. So let's say it adds a C and a G and an A and a T. And again, it's doing that by matching up bases that are complementary to this original parent strand. So this one must have had an A there. This one must have had a T. This one must have had a C. This one must have had a G, and so on. Now, as helicase continues to unzip in this direction, this DNA polymerase can just keep following towards the replication fork. This is called the fork, or the replication fork, so I'm going to indicate that. So this DNA polymerase is moving towards the replication fork. So this side that's being replicated here is called the leading strand. We say that on the leading strand, the DNA is replicated continuously. Because this DNA polymerase molecule can just keep following helicase as it unzips the DNA. Now this is a good time to grab a colored pencil so that you can highlight some things here. I can't really highlight with a smartboard, but I want you to take a colored pencil and I want you to shade in these original bases that were put on there by RNA primase. These are considered the primer, and the primer is made of RNA. Now, usually when we talk about RNA, we're talking about messenger RNA is being made during transcription. So you can see how we've got RNA here, we've split open DNA. This looks a lot like transcription, but it's not. This is replication. When we're done, we're going to have two DNA molecules, double-stranded DNA molecules. We'll have, I'm going to put molecule 1 here. This is DNA. And then when we're done, we're going to have that second molecule. So here's molecule 2 of DNA, one for each daughter cell. Now let's show you how the other strand is being replicated. This strand down here is called the lagging strand. And it's going to be made in pieces or fragments. And these fragments have a name. They're called Okazaki fragments. Now, remember I said that DNA polymerase 3 can only build new strands in the 5' to 3' direction. Well, if we go to build a strand of nucleotides here in the middle, to build that new strand in the 5' to 3' direction, DNA polymerase has to move away from the fork. So close to the fork, in comes some RNA primase, and it's adding the primer. So let's put a primer on there. I'm going to put several U's to help you remember that we're making this primer out of RNA nucleotides. So there's a couple U's there. Let's just put an A and a G. You might want to box this off and shade it in just like you did up here for this one and indicate this is the primer. I'm going to write it pretty small. So here's the primer. And now again we have a 3' end here because this ends 5'. And we now have a place here where DNA polymerase 3 can attach on and it can continue replicating. But again, it has to make that new strand in that 5' to 3' direction. So you're going to see as helicase continues to unzip the DNA, it then creates an area here that can now be replicated. So we'll end up putting a primer here, DNA polymerase will hook on, it'll continue to replicate this section. So you can see that we make this lagging strand in sections or in fragments, and they're called those Okazaki fragments. Now, I'm not quite done yet because here we have DNA, and we have parts of the DNA made of RNA, and that's not going to work. So we have another molecule that's going to come in, and this is the DNA polymerase 1 that I'm talking about. If you can just say there's another DNA polymerase molecule that does what we're about ready to describe, then that's fine. You don't have to remember the 3 and the 1. So what's going to happen is DNA polymerase is going to come in, and it's going to remove these primers, and it's going to replace them with DNA nucleotides, because if we're trying to make DNA, we want the DNA to be made out of DNA nucleotides and not RNA. Whenever it removes that primer, you're going to see here in a different diagram, I'll show you this, there's going to be a gap in the backbone between two nucleotides that are side by side. And so here comes our gluer. Here comes ligase. It's going to come in and it's going to seal up that sugar phosphate backbone. Now, I knew that first diagram was going to get all jumbled up here. So there's more diagrams of replication coming, and we're going to walk through the processes of replication and the different jobs of the enzyme. Now let's look at this next diagram that I'm giving you, and let's just see if you can indicate what some of these different proteins are. Now, I would say that this diagram is not showing you that further down the line we have topoisomerase. So I'm going to add topoisomerase there so we don't forget it. And again, demo in class, that's going to show you what topoisomerase does. I would say this is helicase, unzipping the two strands of nucleotides in the DNA molecule. We have a single strand binding proteins holding apart these two strands of nucleotides there. And we can tell that this molecule is just coursing down, and it looks like we're getting some double-stranded DNA as this new strand, which you see here in the light blue, is being synthesized off of that parent strand, which is the darker strand. So that is your DNA polymerase. And it's 3. You should also be able to look at this diagram and tell that this is the leading strand because it's being made continuously. If I show you this side of the DNA molecule as the 5', you should be able to trace it all the way down to the end and know that at the end of this nucleotide right here, this last one, that is a 3' end. The very end of this nucleotide right here, that must be the 5' end. And you can tell that this new strand, that lighter blue one, is being synthesized in the 5', because it was 5' here, to 3' direction. Now this diagram is showing you the primer at work. So this is this pink one right here, that's RNA primase. And you can see that they're showing you the primers in a different color to indicate those primers are made of RNA nucleotides that are put on there by RNA primase. It looks like this enzyme attached here, because there would be a 3' end on the end of that last RNA nucleotide, and it's moving this way. Again, it's moving in a 5' to 3' direction of that new strand. This is another DNA polymerase 3 molecule. And this is what I mean by those Okazaki fragments. So actually, this one was made first whenever the fork was opening way back there on this DNA molecule. And then this one right here, we can see that it would have been made second. And this is the latest one that's being made here. So this one's being made third. Here's another DNA polymerase molecule that is moving this way after it attached to this 3' end on that RNA nucleotide there. And then we can see a couple more enzymes there. And I should add a box for this one right here. So I'm going to add a box because there was a primer there and it's now been taken off. So there's no red there anymore. It's been replaced with those DNA nucleotides. So this is that other DNA polymerase molecule that I mentioned. This is one. It's taking off those RNA nucleotides and replacing them with those DNA nucleotides. And then finally, you can see that they're showing you there's a break there. So there's a gap there between the phosphate and sugar along the backbone. This gap has to be fixed by that last enzyme we talked about. There's ligase. It's going to seal the gap. Now that you've looked at 2 different diagrams about replication, I would hit pause and see if you can fill in the blanks. To start, we have DNA being unzipped at the H bonds by helicase. We have those single strand binding proteins, keeping those individual strands of nucleotides apart. Up ahead, we have topoisomerase, cutting the DNA as it gets too much tension on it, unraveling it, and then rejoining it together. We have the primer region being added, and that's added by RNA primase. And again, that primer is built of RNA nucleotides, not DNA nucleotides. But that primer gives a 3' end that DNA polymerase 3 can attach to and continue to elongate that new strand of nucleotides. Only one strand is built continuously as the DNA unzips, so it follows helicase as it moves, and that's called the leading strand. We see that it is made continuously. And the other strand is built in segments, and again, those are called Okazaki. And this strand is called the lagging strand. It's being made in pieces. Whenever we remove that primer, then we have a gap that we have to seal there, and that gap is sealed by ligase. And it's actually DNA polymerase 1 that removes those RNA nucleotides and replaces them with the DNA nucleotides. In addition, DNA polymerase is also proofreading for errors, because remember, sometimes the wrong bases can be matched up. And so DNA polymerase is also proofreading and trying to correct for some of those errors. And it might feel like overkill, but again, this is just another diagram of replication, and you should be able to explain what's going on here on the leading strand and the lagging strand, and why we're building these new strands of nucleotides continuously, and why we're building these down here as fragments. You should be able to know that that was RNA primase that's putting on the primer there, and you can see now we have a 3' end that our DNA polymerase can hook onto and continue to synthesize this new strand of nucleotides according to the parental strand here, which is green in this case. And we see another DNA polymerase here, so let's go ahead and we should be able to label those DNA polymerase. We can see there is an enzyme and it's taking off the red RNA nucleotides, and we might want to indicate that those primers are made of RNA. We can see that they're being taken off here, so that must be DNA polymerase 1. And we see ligase is over here and it's sealing up the backbone. Now in class you guys are going to build models, and then you have to explain it to me or your partner. It just depends on who gets lucky and has to explain it to me. We've been concentrating on the specifics of replication. And before I go on to the next page, I do want to emphasize just the big idea behind replication. This is happening to the DNA because the cell has got a signal to divide, and it has to make a copy of each one of its DNA chromosomes. So here is one copy. So this DNA molecule is going to end up going into this daughter cell. And then this DNA molecule, which looks just like the other one, is going to end up being sorted into the other daughter cell. Now that we're done talking about DNA replication, I want to revisit this enzyme called topoisomerase. When we look at this diagram, we can see that DNA is being replicated and it's being unzipped, so this would be helicase. When helicase unzips the DNA, it can cause tension to develop on that DNA molecule. And I'm going to show a demo in class where you're going to see this happen. And that tension can lead to these coils, and they're called supercoils. So this is just showing you, when we see supercoils, that means there's a lot of tension that's being built up on that backbone of the DNA. Eventually, so much tension can develop that the DNA can break, and we have a small little break right there, and that would be bad. We don't want DNA to break. Topoisomerase is going to prevent the buildup of tension. So let's show a molecule come in. So this is an enzyme. This is topoisomerase. And what it'll do is it'll cut the DNA, and it'll unwrap it so there's less tension, and then it will reseal it. So again, topoisomerase relieves the tension so that the DNA doesn't break on its own and have to be fixed. Next, I want to talk about why cells ultimately will stop dividing. Because cells will divide about 50 times and then stop. Even though there's cells around it dying, they'll no longer divide to give you new body cells. And it boils down to this. When the telomeres of the DNA become too short, it's a signal for the cell to no longer divide. So what is a telomere? The telomere is the end of your DNA molecules. So the end of the DNA molecules are called the telomeres. They're just a sequence of bases, but they do not code for any proteins. So down here in this diagram, this is the end of a DNA molecule. So this area of the DNA would be considered the telomere. Now, one thing that you need to know about the telomeres is every time the DNA is copied, the telomeres get shorter and shorter. Now, as to why that occurs, it's not really that big of a deal. That you know that. It has to do with the primers. But just know that when the DNA gets copied, the telomeres shorten. Now, when you're young, this isn't a big deal because you have a gene that codes for a protein called telomerase. So there's a telomerase gene that codes for this enzyme telomerase, and telomerase can rebuild the telomeres whenever you're young. Let me move this down a little bit so that you can see that. So this is telomerase, and it's extending or rebuilding the telomere, preventing it from shortening up over time. Now, whenever you get older, let's indicate when you are older, you no longer make telomerase. This gene becomes inhibited. It gets turned off. So therefore, you can't extend the telomeres. So every time your cells divide, your telomeres get shorter and shorter. And eventually, when the telomeres become too short, which is about after 50 divisions, then that cell will no longer divide. What's interesting about cancer cells is cancer cells can divide forever. So obviously, they're not limited by about 50 divisions. So cancer cells, they keep their telomerase genes active. So they can continue to divide indefinitely. If we could figure out a way to turn off those telomerase genes in those cancer cells, then those cancer cells would eventually stop dividing. Now, on this page, I put some information about HeLa cells. And since we've been talking about cancer, HeLa cells are a cancerous cell that was taken from a lady in the 1950s. And since then, we've been using them for cancer research. Now, on the AP exam, you might be asked to discuss an ethical issue. And this is a good one to talk about because those cells were taken from a lady named Henrietta Lacks without her consent. So this is an ethical issue. Should cells be taken from people without their consent? If money is made on the research that's done on those cells, should it also be given in part to the family, or should the pharmaceutical companies keep all the money that's made off of these cells and so on? So just take a second to learn a little bit about these HeLa cells. And again, you should be able to bring up or discuss an ethical issue related to research.